here she is. Dr. Kimberly Reynolds. Dr. Reynolds is an assistant professor in the Green Center for Systems Biology at UT Southwestern Medical Center in Dallas, Texas. Kim's lab uses a combination of statistical genomics and high throughput experiments to understand constraints on protein activity, abundance, and regulation inside the cell. Her group has used coevolutionary analysis to engineer new allosteric regulation into proteins and to identify metabolic enzyme pairs with constraints on relative expression. So thank you, Kim, for being here, and I will turn it over to her.
Hi, thanks, Leah. Let me quickly screen share. All right, can everyone see my slides okay?
Yep.
Yeah, okay, great, thank you. Okay, so first of all, thank you to the organizers for the invitation to speak here today. It's really a pleasure to be here and be able to share some new science from my lab with all of you. And I'm going to be telling you about some relatively new work mapping the determinants of allostery using deep mutational scanning. Okay, so as many of you know, allosteric regulation really permits the control of protein function from a remote site. And here I'm showing kind of three examples of different allosteric mechanisms, right? So you could have allostery that works through something like small molecule ligand binding. So for example, here there's an allosteric inhibitor that modulates catalytic activity over here. You can have allosteric regulation that works through post-translational modification. So you could have like a phosphorylation or some other kind of PTM that comes in and then modifies activity. Or you can also have allostery that is modulated through protein-protein interactions, right? So in this case, this CDC42 GTPase comes in and binds to this little PAR6 PDZ domain, and it's known that this binding modulates peptide binding affinity over here. Okay, so in all of these cases, the kind of salient feature that they all share is that you have some kind of remote effector, whether it be a small molecule or a protein ligand, that comes in and modulates the conformational landscape and or kind of thermodynamics of some output protein such that it changes its activity. Okay, so allostery is really a very common feature of proteins, and it can occur at different sites and by different mechanisms for different homologs. And here I'm showing the protein kinases as one kind of prototypical example of this, right? So what I'm showing here in panel A is just a canonical protein kinase structure. There's an N-terminal lobe, a C-terminal lobe, and an active site here. And then what I'm showing in panel B is now I've taken this kinase structure, and on the surface, I'm painting in different colors, different sites of regulation in different kinase homologs, right? And so as you go through and start to add on different regulatory sites from different kinase family members, this thing starts to quickly look like kind of an Easter egg, right? So for example, you can see that like the cyclin-dependent kinases are regulated kind of over here on this blue surface. Some kinases are regulated by dimerization on kind of the back side of the kinase, right? And you quickly get this view that, allosteric regulation can happen sort of all over the place. And, that led to, I think, a really interesting question that was very nicely posed by Kurian and Eisenberg in this 2007 perspective piece in Nature, right? And, the question is really, how does all of this regulatory diversity evolve, right? Because in many ways, like if you think about allosteric regulation, it feels like kind of a molecular scale instance of, quote, irreducible complexity, in the sense that you have some remote surface that controls the active site, and presumably there have to be a lot of kind of molecular parts in place for that, allosteric regulation to work, this coordinated action of amino acids. But in practice, when we look at natural systems, they evolve allosteric regulation all over the place. So despite the fact that it, seems like it might be kind of a challenging feature to evolve, natural systems don't just use sort of one allosteric surface over and over again, but they actually evolve sort of a diversity of surfaces. Okay, so we wanted to try to understand how this allosteric regulation might evolve. And in doing so, the goal is to provide some kind of insights into how we might engineer or build allosteric regulation in the lab. Okay, so we're going to start with an idea that's kind of coming from statistical analysis of protein sequences, right? And so here the basic idea is if we take a bunch of natural protein sequences, so we take homologs of a given protein, and we look closely at their amino acid sequence, perhaps we can learn or infer kind of the rules for constructing these allosteric systems. And to be more specific, the idea is that we want to infer interactions between amino acids based on their kind of coevolution across homologs. So the general scheme of these analyses is that we start off with a multiple sequence alignment, where we're starting with hundreds to say thousands or now even tens of thousands of sequences. So these are all what we're presuming are homologous sequences based on sequence identity. And then we start looking at the conservation and particularly the correlations in conservation between pairs of amino acids, right? So for example, if I'm looking at positions 1 and 2, I can see that every time this like pink residue changes to purple, then I get a correlated change at the second site from yellow to green. And then based on that, I can infer, well, maybe these first two amino acids are subject to some joint or shared functional constraint, right? And, you know, very loosely speaking, perhaps this means that they are kind of talking to each other. Okay, so now you can go and make this kind of statistical measurement across the entire length of the sequence and construct a covariance matrix here. So we go from 1 to the length of the protein in positions from 1 to the length of the protein. And we get a square symmetric matrix where now each entry is telling us about the kind of correlation strength between positions, and what we're inferring is kind of coevolution of these sites. Okay, so in practice, what does this look like, right? We have our multiple sequence alignment, we calculate our covariance matrix. And when we start to look at these, one thing that seems to be true in a diversity of protein families is that most positions in the protein don't have a strongly correlated or coevolutionary signal, right? So in this matrix, blue indicates sort of very little correlation, and red indicates a higher degree of correlation, or as we're inferring, coevolution. And so you can see that while there's a handful of positions that are really strongly co-evolving, the vast majority of this matrix is blue. Okay, and so now we can take this matrix and we can analyze this matrix to infer groups of residues that are strongly co-evolving with each other. In practice, we do this using principal components analysis, right? And we can take those groups of co-evolving residues and map them onto the protein structure. And these groups of collectively evolving residues, I'm going to refer to as sectors here and throughout the talk, right? And I'll show the sectors as these kind of blue positions that are painted onto the protein. Okay, so now in several studies that have been, several protein families that have been studied, what we've seen is, you know, sectors seem to empirically have these kind of three physical properties. The first is that they're relatively sparse. So it's usually about 20 to 30% of the protein sequence that's really strongly co-evolving. And they form these kind of physically contiguous networks that are branching throughout the protein structure, right? So here I'm showing 3 examples, a PDZ domain, a serine protease, and dihydrofolate reductase. And at the bottom, what I'm doing is showing cut throughs so you can see how the sector is distributed inside the interior of the protein. And you can see, for example, like in dihydrofolate reductase, this enzyme, that the sector is kind of centered around the active site. And then it has these branching arms that seem to extend out to these distal surfaces on the protein surface, right? And this is also true in the serine proteases and in the PDZ domain. Okay, so kind of looking at this architecture across protein families generated an idea, which is maybe what the sector is doing, or could be seen as a way to kind of wire, you know, quote, wire up multiple protein surface sites to the active site, right? So here again, I'm showing the distribution of these sector positions in the interior of the enzyme dihydrofolate reductase. And you can see that it's connecting up these kind of distal surfaces to the active site. And so the hypothesis is, well, if a sector really represents kind of a cooperative group of amino acids that are communicating with each other, then perhaps what it means is now if I push on this little surface site here that we know co-evolves at the active site, that potentially this is kind of a preferential site to actually modulate catalytic activity. And if I were to somehow manipulate or push on a surface over here in gray, that I would be less likely to change activity of my protein, right? So maybe these sites that are these sector connected edges can act as kind of preferential sites or hotspots for introducing new allosteric regulation. Okay, so in prior work, I set out to test this. And the idea was to basically start with 2 protein domains, right? So this is kind of the abstract idea. We're going to have some output domain where we can measure activity. And then we're going to plug in an input domain, right? That's going to be our allosteric input. And the idea is if we connect these 2 things such that the input is connected into the sector of the output, with some probability, we should get these 2 domains to now start to talk to each other. Okay, and so here's what the experiment, the implementation of the experiment really looks like. So we've got 2 domains. As our input, we're taking LOVE2, which is a little blue light sensing domain from plants. And then we're taking this blue light sensor and we're attaching it to this output domain, dihydrofolate reductase. And the idea is if we attach it like this, where it's not plugged into the sector, we're going to anticipate that we'll see no regulation. And then if we're going to attach it like this, so that we're hooked into the sector, we expect that with some probability, we're going to start to see a light controlled enzyme, right? Okay, and so here was the result of this domain insertion study. So I went through and inserted LOVE2 at 72 positions on the surface of dihydrofolate reductase, right? And the red positions are the sites where we actually got light-based control of DHFR, right? And you can see that these places where we got light-based control of DHFR really kind of nicely coincide with these sector-connected edges on the surface of the protein. And in fact, we never saw light-dependent regulation of DHFR at a non-sector site, right? So this case never led to regulation. And then we did see regulation at about 40% of these sector-connected surfaces, right? So not perfect, but with some probability you get light-based control. And this result was statistically significant over a range of cutoffs for either defining these sector positions, these co-evolving positions, or a range of cutoffs for the significance of light dependence in our experiment. Okay, so we see this result. And based on these data, the idea is that we're proposing that proteins contain this kind of conserved cooperative architecture that we're calling the sector that can result in latent allosteric hotspots, right? So the idea is, when we look across a protein family with many homologs, and we see that it has a bunch of different regulatory mechanisms, whether it be regulation by post-translational modifications, PPIs, dimerization, that potentially all of that is actually federated by some kind of conserved underlying architecture that lets you use these different surfaces for allosteric regulation. And if so, then this could be a vehicle to kind of rapidly or facilitate at least rewiring and diversifying signaling systems. But it might also be kind of a liability because now you could sort of spark spurious or off-target regulation fairly easily. Okay, and in a sampling of natural and engineered systems, this principle seems to kind of loosely hold, right? So here I'm just showing like a handful of examples. So for example, in the CDC42 site, you can see that the nucleotide binding site here in red is connected through the protein interface to the PDZ binding site by this kind of sector connection here. In HSP70, a chaperone, we again see that there's a sector that seems to connect the ATP binding site, which is the allosteric modulator, to the substrate binding site over here. We see something similar in the caspases, and then this is also true in a couple of engineered systems, the DHFR system that I just showed you. And then also in cathepsin, another group actually engineered a cathepsin inhibitor based on the idea that they were going to target these sector connected sites. Okay, so we're proposing that the sector provides a substrate for evolving new allosteric regulation or at these conserved surface sites. And the question is like, what are the next steps, right? So once you initiate this allosteric effect, we wanted to know how and where can you optimize regulation, right? So one thing that I didn't tell you on the previous slides is when we made these DHFR LOVE2 fusions, the initial allosteric effect that we saw was really modest, right? So some of our, kind of biggest engineered allosteric systems changed activity by say 20% upon light activation, right? So the catalytic activity would be 20% greater in the light versus the dark. And I think this is a level of allostery that most biochemists would find really quite middling, right? We're not seeing sort of huge effects. And so the question is like, okay, if this is really kind of a viable evolutionary starting point, can we now take this and optimize this through a path of kind of single mutation and variation to show that this would be kind of a useful substrate to build a real allosteric switch with? Okay, so specifically, we wanted to ask, first of all, is the pattern of mutations that can influence allostery in these systems sparse? Is it just a few mutations, or is it really abundant? Secondly, is the pattern of mutations that influence allostery localized? Like, for example, is it all around kind of the effector site, or, in this case, is it all localized around the kind of domain insertion site, or is it distributed throughout the entire protein structure? And then finally, is there any relationship between the mutations that influence allostery and the sector, this group of co-evolving residues that we identify through sequence analysis? Okay, so to address these questions, we set out to do a deep mutational scan. And the work that I'm about to show you was really led by James McCormick, who's a postdoctoral fellow in the lab. And he had assistance from Samuel Thompson, who actually constructed our deep mutational scanning libraries. Samuel at the time was a graduate student with Tanya Kortem and has now gone on to a postdoc at Stanford. And then Max Russo, who's a research assistant in the lab, did a lot of the kind of protein purification and biochemistry work that I'll show you kind of towards the end. And this work we just put up on bioRxiv relatively recently. So if you're interested in it, please go read more. Okay, so here's our model system. As I told you before, we did this kind of domain insertion scan using LOVE2, this blue light sensing domain, and dihydrofolate reductase. So we're going to pick one of these fusions that has a modest allosteric effect, right? And so the way the system works, we call the system DL121 for DHFR LOVE2 position 121, because LOVE2 is actually inserted between residues 120 and 121 of DHFR. And the way LOVE2 works is that there's an FMN chromophore that actually absorbs a photon of blue light. And then in response to that photon absorption, this J alpha helix, at least in its native context, is thought to kind of unspool. And so you get this kind of folded to unfolded transition. And then for the purposes of our experiments, it's really convenient that the N and C termini are really close together. So what we're making is just a chimeric protein fusion where these N and C termini are plugged into the peptide bond of position 121 on dihydrofolate reductase, okay? DHFR is an essential metabolic enzyme that reduces folates. And this is very useful for us because then it's reasonably straightforward to link its activity to growth rate. And I'll talk about that a little bit more in a moment. Okay, and so as I told you, this is has a very kind of modest allosteric activity. So we see actually a 28% increase in kcat in response to light. So it's modestly light activated. Okay, now because DHFR is an essential metabolic enzyme, we can link its activity to growth rate, and this really forms kind of the crux of our assay. So what we're going to do is we're going to measure growth rates for our mutants in the light, we're going to measure growth rates for our mutants in the dark, and then we're going to subtract the two. And in prior work, what we had seen is that for DL121, this 28% difference in catalytic activity translates into about a 20% growth rate difference in the light. And here I'm showing this in comparison to a C450S control. So this is a mutant of LOVE2 that no longer is light responsive. And then a handful of DHFR point mutants that do not contain a LOVE2 domain, right? So you can see that we get sort of a modest growth rate difference and a modest catalytic improvement in response to light. Okay, so the idea here is we're taking a system where the input signal is rapidly applied. We can put light on. The allosteric effects on activity can be quantified both in vitro biochemically and in vivo in our E. coli-based assay. And there's a potential for large improvements in allosteric dynamic range. So what we really wanted to see is, we expect to see some mutations that disrupt allosteric activity, but where can we get improvements in allosteric activity? Okay, so here's our overall experiment. We're going to take our DL121 fusion, which is sector connected and weakly allosteric. We're going to do saturation mutagenesis or deep mutational scanning over the entire DHFR domain, right? And the idea is we want to measure kind of which DHFR positions are coupled to this light-based input. And the way that we're going to do this is we're going to measure the relative growth rate of every mutation in the light, of every mutation in the dark, and then we're going to subtract these two to look at kind of the allosteric effects of these mutations. And so to do this, from the outset, we wanted to make sure we had a assay with really good resolution because we're expecting that many of the mutational effects will be small, and we want to be able to see these, right? So here's kind of how we fixed up our assay, right? So we're doing our deep mutational scanning assays inside of a turbidistat. So here I'm showing a picture and a little schematic of the vials that we use to do these experiments. So our turbidistat construction is based on a morbidistat design from Erdal Toprak and Roy Kashoni. And so basically we have this platform, this stirring platform housed inside of an incubator. We have an array of 15 separate vials, or up to 15 separate vials. We've put in blue LEDs to trigger the LOVE2 domain. And each culture holds about 15 mils of volume, where we're dynamically sensing the optical density by an IR LED detector emitter pair. And then we add in fresh media in response to that OD detection, right? So the way these experiments look is we can inoculate our cells in, they grow up, and then once they hit this target OD, we get these cycles of grow, dilute, grow, dilute, grow, dilute. And the idea is that this is going to give us kind of systematic control of our media conditions and growth state. So we don't have to do kind of serial dilutions, and we know they're growing under relatively constant conditions. Okay, and now we're going to do the standard demutational scanning thing. So we're going to take time points and we're going to sequence to get allele frequencies, right? So in these experiments, we actually take a series of time points every 4 hours over a time period of about 24 hours. And here I'm showing the data for wild type DHFR, DL121, which is our DHFR LOVE2 fusions, and then a handful of DHFR point mutants that lack a LOVE2 domain, but where we've characterized their catalytic activity, right? So our goal here is to first kind of set up a, almost like a calibration curve relating DHFR activity to growth rate. And again, on the y-axis, what I'm plotting is the number of each mutant relative to wild type, and then normalized to t equals 0, right? So wild type is necessarily flat over the kind of duration of our experiment. Okay, and then we see that under our assay conditions, we get a kind of near linear mapping between relative growth rate and catalytic activity over about 4 orders of magnitude. You can see that eventually this mapping does saturate, right? So once we get within in a certain range of the kind of the wild type construct, we really lose resolution. And then our DHFR LOVE2 fusion is sitting right here, kind of in the middle of this dynamic range. And I do note that the DL121 construct is sitting off this calibration line. We think this is because it has like a little bit lower expression potentially than these other point mutants. But I want to emphasize that because all of our measurements are relative, right? We're considering growth rate in the light versus growth rate in the dark. We expect that any kind of changes in expression, as long as they're not light dependent, should normalize out in our assay. Okay, and I'll note here that one of the things that I was excited about when James first showed me this calibration curve is that we can resolve changes within kcat over km given our error of about 0.02 per micromole per second, which is on par with or actually exceeds a lot of the kind of in vitro measurements people make. I think because kind of these frequency sequencing based approach can be so precise. Okay, so we use this measuring out of assay to measure relative growth rates for all of our DL121 mutants in the dark and in the light. And first, I'm going to show you the data for mutations on growth in just the dark, right? And so here we're not inferring anything about allostery, but our goal is just to kind of first look at the data and make sure things make sense and that the things that are deleterious are, you know, kind of expected, like conserve things in the active site, right? And so here again, deleterious mutations are going to be shown in blue, near wild type is white, and then red would be things that are beneficial or enhanced growth rate, right? And so you can see the vast majority of our mutations for this construct were deleterious. In particular, these navy blue pixels that I'm labeled null are things where we couldn't get, the construct disappeared from our library before we had measured 3 time points. And so we couldn't really fit a reliable growth rate to those. So we just label those null and we assume that they're among some of the most deleterious mutations in our library. Okay, so, and the mutations we see are largely deleterious relative to wild type. And you know, I think this is because we're starting from a construct where we've inserted this LOVE2 domain. And so it's, you know, maybe not as robust as its kind of wild type counterpart. And so many of the mutations acting on top of this are deleterious. Okay, so then we wanted to look and see where are the most deleterious mutations happening? Are they places that make sense to us given the kind of known biochemistry of DHFR? And we're going to take this mutation DL121D27N as an empirical cutoff. So D27N is a mutation in the DHFR active site that's known to make it near catalytically inactive. We know from separate growth rate measurements that it's kind of minimally viable in our system. And we're going to take everything to the left of this line and we're going to say, okay, these are all really deleterious mutations. And we see that we get a statistically significant enrichment of these deleterious mutations in sector positions. We also see a statistically significant association between these deleterious mutations and evolutionarily conserved positions, right? So, you know, this is kind of consistent with the view that the sector is critical to protein function. And it also gives us some confidence that our assay really is reporting on kind of important determinants of DHFR activity. Okay, so now we want to go on to analyze allostery, but first we're going to remove these slow growers before we start analyzing allosteric effects. And we do these kind of for 2 reasons. The first and probably the most fundamental one is that inactive mutants by definition are not allosteric, right? So if the protein's dead, you can't really regulate the activity. So we're going to remove these low growth mutations. And I'm showing here that this, you know, really kind of still leaves a good distribution of mutants between kind of common structural or evolutionary categories, right? So we're showing surface positions, core positions, sector positions, and conserved positions. After filtering out these mutations with low growth, here's kind of the fraction of the library that they compose, right? And these are the raw numbers of mutations left after filtering. Okay, and the other thing that this does is it eliminates some of the noisiest measurements from consideration, right? So we don't really expect to even be able to calculate a reliable estimate of allostery for some of these really slow growers. So here I'm showing our 3 biological replicates we did of this experiment. And what we're filtering out is everything in this kind of lower left corner below these cyan lines here. And then these are the mutants that are left that we're going to calculate allostery for, which is about 51% of our data. Okay. So for all of our mutations, we have triplicate, lit, and dark measurements. Here I'm showing 3 representative curves. One is for D27N, our mutation that we know is highly deleterious, and so you can see it's very deleterious both in the light and the dark. We have D122W, which is a little bit beneficial in both the light and the dark. And then we have mutations like E154R, which actually seem to have differential effects in the light versus the dark, right? So here you can see that the growth is a little bit decreased in the dark, and in the light, it's remaining about the same. And so I should point out kind of 2 things. First of all, in all of these measurements, we're normalizing against the parent DL121 construct, right? So what that means is that if you were to see no change in the slope at all, you're actually preserving the allosteric effect of the starting construct. And then the second thing is what we're going to do is we're going to take this slope, and that's what we're actually calling our relative growth rate. Okay. So now we want to go through and kind of assess where do we have a significant allosteric effect for which mutants, right? And so we're going to assess this using a chi-squared test because we have these 3 biological replicates. And I'm showing these data as a volcano plot, right? So on the y-axis, I have the minus log of the p-value. So things up here are more statistically significant. And on the x-axis, I'm showing the growth rate difference. So things on the right side of the graph are allostery enhancing, meaning they grow better in the light than in the dark. And on the left side of the plot, I have things that are allostery disrupting. So they grow worse in the light than in the dark, right? And I'm showing 2 p-value cutoffs. One is a standard p-value cutoff of 0.05. And the second is a p-value cutoff that we've adjusted for multiple hypothesis testing. That's a more stringent value of 0.016. So the results that I'll show you today are with this more stringent p-value cutoff, but I'll say that the results at the more standard p-value cutoff are really entirely consistent with what we see at 0.016. And then these red mutants are things that we actually chose to go express and purify in the lab, and I'll talk about those in a minute. Okay, so in total, looking at these mutants, we observed 69 significant allosteric mutations, which is only 5% of our library, right? So the vast majority of mutations that we measured had, no detectable impact on the kind of starting allostery of the DL121 construct, right? 56 of these are allostery enhancing, and then 13 are allostery disrupting. And here I'm just showing the relative growth rates in the light and in the dark for all 3 biological replicates across these allostery enhancing mutations and then these allostery disrupting mutations over here. So one thing we noticed was there was not really a strong correlation between, say, allostery enhancing mutations and the overall effect on growth rate, right? We saw things that enhanced allostery that enhanced growth, and we also saw things that enhanced allostery that depressed growth rate. The other thing that we notice is many of these allosteric effects are quite small, and I'll show some biochemical data on this in a moment, but we're getting changes of maybe 1 or 2-fold enhancements in allostery with these single mutations that we're introducing, even though we're seeing kind of a wide range of effects on overall growth rate. Okay, so then we wanted to make sure that our in vitro measurements of allostery are kind of correlating to what we're inferring in vivo. And so we went and expressed and purified and biochemically characterized these through steady state Michaelis-Menten kinetics. And what we see is we get a really, I think, pretty good correlation between our in vivo measurements and these in vitro measurements. And the mutant that we have with kind of the largest allosteric effect is this mutation H124Q, which now increases our regulation to 2-fold. So I'll remind you, our starting construct had a 28% increase in regulation in response to light. And so H124Q increases this to about 2-fold. Okay. So finally, how are these mutations structurally distributed? We found that the allostery disrupting mutations, we have 13 allostery disrupting mutations, and these localize actually to 6 positions on the DHFR structure, right? And the allostery disrupting mutations we can see are all kind of proximal to this 120 insertion site between 120 and 121, where we're putting LOVE2, with the exception of this position 34. So in fact, if you plot the fraction of mutations as a function of this distance cutoff, we can see that the vast majority of our disrupting mutations are within this 10 angstrom cutoff. Now, in contrast, the allostery enhancing mutations, this was sort of, a counter to our expectations, but it's distributed across the protein surface, right? So the allostery enhancing mutations are in light blue, and you can see that, we have some near the insertion site, but we also have some of them near kind of the backside of DHFR. There's a strong association between these allostery enhancing mutations and the surface. Okay, and finally, we wanted to ask, how are these, what's their association or not with the sector? So here I'm showing a little movie. The sector positions, again, are in dark blue. The allostery enhancing mutations are in light blue, and the allostery disrupting mutations, again, are here in orange. So you can see here's the insertion site, and then these allostery enhancing mutations are all really kind of peripheral to the sector. And in some cases, they don't even contact the sector, right? So we have some in this loop that's around position 80 in DHFR that aren't contacting the sector, and some down here, which are also not contacting the sector. So in fact, when you look at the distribution of allostery enhancing mutations relative to the sector, we see that we have no enhancing mutations in the sector. And we see kind of a modest enrichment of allostery disrupting positions in the sector. So overall, this gives us kind of this picture that we can start to optimize or tune allostery by variation at kind of these distal and weakly conserved surface sites. Okay, so to summarize, basically what we've seen in past work is that these sector-connected surfaces are hotspots for introducing new allostery. Mutations that disrupt function are enriched in the sector, right? So when we make mutations in the sector, they tend to kind of disrupt DHFR activity. And then while single mutations can exert a large effect on function, the effects on allostery tended to be comparatively modest. A small fraction, about 5% of these mutations modulated allosteric regulation. The allostery disrupting mutations were kind of localized to the domain insertion site, while the allostery enhancing mutations were kind of all over the place on the protein surface. Okay, so I wanted to ask if we could, the effects we're seeing are still pretty modest. So can we start to combine these single mutations to yield sort of a higher performance switch? And so to start with, we've picked kind of 3 mutations, D87A, H124Q, and M16A. Individually, these all have kind of allostery enhancing effects. And then when we combine them, we see that they actually combine kind of consistent with log additive expectation, right? So they seem to be kind of acting independently to tune allostery, at least for this subset of kind of 3 mutations. So that's kind of exciting to us that they seem to combine very nicely. And so maybe this presents a possible path for starting to evolve or optimize allosteric regulation, where you have kind of an introduction of allostery through an initial domain insertion at one of these sector connected surfaces, right? This could be like a transposition or a recombination event. It's important to say that domain insertions are also common in natural systems, not just engineered systems. And that this could then be optimized through some kind of surface residue variation. And so now this is now something that we're thinking really hard about. How can we watch this process in the lab and see kind of multiple steps towards an enhanced allosteric system. And with that, I'll wrap up. I'd like to thank the people in my group who did the work. So James, Samuel, and Max. Thank my lab, our collaborators, our funds, and welcome any questions. So thank you.
Thank you, Dr. Kimberly Reynolds. That was a really awesome talk. You have lots of questions, so we'll go ahead and get started. So Leah Starita asks you, what do you think is special about the 40% of sector-connected sites that allowed light regulation of DHFR versus the ones that did not?
Yeah, that's a great question. I'm not sure that there's anything really like special about them at all, actually. I think that it has to do, my best guess, I should say, is that it has to do with some kind of local sterics or geometry of like plugging LOVE2 into that particular surface site, right? It could be that if we repeated that domain insertion scan again with a slightly different linker length or with a different domain, that we would expose like a different set of surface sites that were allosterically responsive. That's what I would guess. But of course, we haven't done the experiment, so yeah.
Awesome, thanks. And then Justin Kinney asks, how was the DHFR library constructed, and did it contain any double mutants or higher order mutants?
It only, well, to our best knowledge, it was limited to single mutants. So we constructed it through inverse PCR with NNS primers. So Samuel went through and sort of did a PCR at each site in DHFR. And then we spot checked the library by Sanger sequencing a handful of those constructs through the entire DHFR coding region to verify they were single mutants. We did break the library into 4 sections to make it amenable to sequencing with like a 300 cycle kit. So there's basically 4 sublibraries that represent each quarter of DHFR. So if there were somehow a double mutant outside of that region, we wouldn't have seen it. But we consider them, probably fairly unlikely events given how the library was made.
Mm-hmm. Okay, and then Willow Coyote Maestus asks, how do you think the dynamic ranges of mutations within DHFR would compare to optimizing linkers to connect LOVE2 to DHFR?
Yeah, that's a great question. We haven't done any work on linker optimization. So I do wonder if we kind of tuned up the linker region, if we could sort of get immediately kind of a more enhancing signal. I think it would be worth doing that experiment. I'll say our goal here was we really wanted to focus on single mutants from kind of this evolutionary perspective. But I think that is a good question because the kind of engineering strategy is usually like, if it doesn't work, you fiddle with the linker. So it's a great question.
Okay, and then Elizabeth Brunk asks, of the 69 mutations, do you see a trend in variant effect versus amino acid type?
Not to our best efforts. So far, we don't really see a strong trend with any particular amino acids types. We looked a little bit, we were kind of interested to see if maybe it could be something like glycines or something that modified like backbone entropy might modulate allostery, but we did not see anything statistically significant, in our analysis so far. So.
Okay. That's pretty interesting. Okay, and then Juanan Zhao asks, how do you differentiate between mutations that affect allostery from those disturbing protein stability?
Yeah, so we're looking for things that are, I guess, having differential effects in the light and in the dark, right? So I guess we would see if there were a mutation that had differential effects on stability in the light versus dark, that would look like an allosteric mutation in our assay, right? So in that sense, we're not really differentiating between something that has a direct effect on stability versus activity. That said, it would again have to be a mutant that has a differential effect where it affects stability, differently in the light versus in the dark. So far, the mutants that we've characterized in vitro, the effects seem to really be on Kcat. We didn't really see a lot of effects on Km, the Michaelis constant. So we think a lot of them are acting through catalytic activity, but it'd be worth looking at stability for some of these as well.
Yeah, that makes sense. The next question is from Doug Fowler, and it's one I'm interested in too. So he says that the best allostery enhancing mutant increases light responsiveness by 2-fold. That's still pretty modest. Does that represent a maximum, or do you think that there are constraints on allostery, possibly big ones that can't be resolved by single mutants?
Yeah, I think this is a great question. So, I mean, one thing that we were encouraged by is when we start to combine these, we can get up to a 4-fold allosteric effect, suggesting that, we're not really at a ceiling. We know that in natural LOVE2 containing proteins, the allosteric effect of LOVE2 can be much larger than, kind of the few-fold that we're seeing. So I don't think that we've kind of tapped out the allosteric potential of LOVE2 necessarily. I do think it's a good question of kind of, clearly we didn't see a whole lot with single mutants here. It would be interesting to compare this to single mutations affecting allostery in a natural already tuned system. And then, in future work, we're now planning to do some higher order mutagenesis and combinations of these mutants to see how good can we do in this particular system. So.
Yeah, that'd be super awesome to see.